And in the next 20 minutes or so, I will tell you why I don't think so, and I will tell you why data don't really show that there is much improvement. First, there are three flawed concepts of the contribution of molecular findings when it comes to the diagnostic accuracy of morphologic diagnosis. The first one is the concept of the dysplastic nevus as a precursor of melanoma, and you heard a lot about this, so I will just very briefly touch it. The next one is the concept of histopathology as a gold standard. This is something that has been true in the past, but isn't at the moment, and also here, thanks to Harold, I don't have to give you very long explanations, but just a short overview. And the rest of the talk will be the problematic concept that we think that genetic alterations will reveal themselves in a morphologic diagnosis, so a name that we see. And we come to this later. So the concept of the dysplastic nevus as a precursor, and this is how it is seen. As we heard, most of the melanomas develop de novo, so not from a nevus. So it's melanoma from the onset. And our problem is just that morphologically, is there a pointer? Morphologically, there is this area where it's just difficult for us to assess either by dermatoscopy or by clinic or by histopathology whether it's benign or malignant. The concept of histopathology as the gold standard or morphology as the gold standard was true at a time when lesions like this one, which today will be, without any doubt, will be diagnosed as malignant, as melanoma in situ, were considered to be benign, precursor lesions, melanocytic dysplasia, lentiginous type. And the diagnosis of melanoma was made when lesions were this size. They were big, irregular plaques, ulcerated, and biology told you that you were right because, like here in this patient, you already see the lymph node metastasis. And this is what supervised learning means. Biology serves to assess the meaning of a morphologic finding. But fortunately, today we remove lesions much earlier, but as a consequence, we lost biology, and you heard about this problem, and I think that's the main problem of all the problems that follow from that. And in the lack of biologic information, we have now morphology as our gold standard, and morphology just fails us in problematic lesions. So the hope was that if we introduce molecular findings or the diagnosis of molecular findings into this problematic area, that we will improve our diagnostic accuracy. There are several different mutations that play a role in the initiation of human cancer. They are roughly divided into inactivating intragenic mutations and activating mutations, gene deletions of tumor suppressor genes, epigenetic silencing, so when regulatory regions are affected, gene amplification of oncogenes, and also gene translocations when there is a fusion of DNA fragments from different chromosomal sites. These are all alterations that ultimately play a role in the development of melanoma. And I will not go into detail now in pathways. It's the MAP kinase pathway that gets activated and that plays the main role. But I will go right into the methods that are used to assess or to resolve the dilemma between benign and malignant. And I'm going to discuss these three methods and start with the comparative genomic hybridization. So this is a method that uses either fresh or bulk frozen or paraffin embedded tissue. And what you do is that you have your DNA of interest, so the tumor DNA and the control DNA. And you label your two DNAs with different fluorescent colors, so usually red, the tumor, and green, the normal DNA. And in conventional CGH, you hybridize these probes to metaphasic chromosome spreads. A development of this conventional method is the cDNA microarray. The results that you get is that if you have gains, chromosomal gains, then you get a green result, like a red result, sorry, like here. If you have losses, a green result. If you look in an array, it gives you a real fluorescence image or map. and a nice picture that you can read out. So what are the advantages of this method? You can identify the chromosomes that carry the aberrations. You can analyze large amounts of targets without prior knowledge where the alteration is located. You do not need specific probes as is necessary in fish. And you can detect the presence of amplified genes, and you can draw more or less a map of where these genes are located. The problems is that this method is prone to inaccuracies if you have certain regions affected, like repeat sequences or telomeres, so when the reading gets more problematic. But also chromosomal gains or losses that are balanced will not show up in your result because there's a problem here. Another thing that you must have to be aware of, that you need to see your chromosomal changes in more than 50% of cells. And so if you have changes in very early lesions that just affect a lower percentage of cells, you will miss it. And contamination of test cells with normal cells, of course, is a problem. And the last problem in practice is, of course, that it's a quite expensive method to do, and you need a lab and you need facilities. Boris Bastian had done this analysis in 32 cases of primary melanomas, and he found certain chromosomes that are more likely to carry these alterations. So you see in green chromosomes where there are gains, in red where there are losses, and it turned out that chromosome 6 and chromosome 11 are good candidates if you look at samples where chromosomal changes occur. The next method, fluorescence in situ hybridization, FISH, is what is heard about a lot and published a lot. And how does the method work? So you have your DNA probe and you have your target sequence. The DNA probe is labeled with either directly or indirectly with a fluorophore. Then you proceed to get single-strand DNAs, and in the hybridization process you have the binding of complementary DNAs, and finally analysis by fluorescence microscopy. Why is this a nice method? Well, the advantage is that you can visualize copy number changes and translocations, and you don't only have your genetic information, you also have a morphologic information. So you have cytogenetics and histology linked, because you can do it on paraffin-embedded sections. The problems is that it's really sometimes or always difficult to interpret, because in a section that is on a paraffin-embedded section on a slide, you have a lot of incomplete nuclei, and these are signals you find there are difficult to interpret. Also, you can only look at a certain number of locations in one sample, and this is at the moment limited to 4, and we go into this problem a little bit later. And in small lesions, again, you have the problem that you might not get a result, because you need to assess between 100 and 200 nuclei to come to a meaningful diagnosis. So in small lesions, you might not find this number of nuclei. The FISH probes that are used for distinguishing nevi to melanoma, and again, here it's just interesting to know they are all 3 of them on chromosome 6, 1 on chromosome 11, so either suppressor genes or oncogenes. That's not the important part. What is important, here you have an image how this method looks. So if there are no aberrations, you have diploid signals. If there are alterations, then the number changes. This is quite interesting because it shows